Welcome to the second week of the lectures for the course Human Molecular Genetics. In the previous week, we looked into the concept of central dogma of molecular biology, where we looked in the genetic material, what are the experiments that led to understanding as to how the genetic material function, how the information is processed, and how defects in your genetic material can have an effect on the cell, system, and organism. So, today what we are going to look into is how we look into the human population or the family and understand the phenotype that you see, which is resulting from a defect in the DNA. We can track them as to what kind of inheritance that is running through the family, the characters that run through the family, what kind of inheritance are there, and how that information can be used to predict the onset of such diseases in the future generation, and also to identify the chromosomes that could have the defect and eventually the gene characterization and so on. So, we will start with a concept called as pedigree analysis, wherein we talk to individuals that are affected with a given disease, or at times it may not be a disease, but a phenotype that is very different from a normal population, and then see how it segregates in the family. So, this is typically called as pedigree analysis, and that is the concept. So, let us look into one simple concept. What do you mean by phenotype? A phenotype could be any character that distinguishes certain individuals from the rest of the population. One classic example is hand clasping. When you close your eye and you clasp your hands, you would find always the majority of us we will have the left thumb going over the right thumb. This is considered to be wild type, meaning the majority in the population would have such clasping behavior. But if you do it in alternate way, you will find it is difficult. You try it out, you close your eyes and try to close it, you will find putting your right over left, you will feel it is uncomfortable, right. So, that is what is shown in this slide here. The left over right is considered to be a dominant character, whereas the right over left is a recessive character, but still you see there are good number of individuals in the population who have this character. That is, again, we believe that there could be genes that are underlying such character. So, this is one such character that you see. So, this perhaps you can even trace it in a family, and that is how you call it as one, the right you call it as dominant, because you can trace it in the family and that is being inherited. That is with regard to a character which not necessarily affect your normal life. It does not really matter the left comes over the right or the right thumb comes over the left. You are all right, you can play cricket, you can cook food, you can do your studies, absolutely it does not affect. But at times there could be other characters which could affect your survival. For example, you are blind, and that could be because of some genetic defect that are there in your DNA, and that would affect your survival. Or you may not be able to hear, hearing impairment, or you could have other problems which again run in the family because the defect originates from a defect in the DNA. Therefore, the DNA is transmitted from one generation to the other. You could trace it in the family, right. So, that is how do you really do that. Let us look into the classic way. So, whatever probably you have studied in your textbook so far, we talk about the Mendelian genetics, and more often we call also some of the disorders in the human as Mendelian characters, because the whatever laws that Mendel proposed, it applies even to many of the genes that and the phenotype that you see in the human. One of them, of course, is the law of dominance, wherein he suggests that one particular trait is dominant over the other, and if you could remember that he has used certain plants and certain characters for proving that hypothesis, and we call it as a law because it now majority of the genes obey that. And for example, what is shown here is the flower color. Let us assume that the orange or red color of flower is dominant over the yellow, and we can pretty much test whether the dominant phenotype that you see, the orange color, is because of a particular genotype. For example, whether that color is because of a heterozygous condition, only one allele is dominant, other one is recessive, or it is homozygous for the dominant allele, that is both are both alleles are for the dominant phenotype, we can test that by doing what is called as a test cross, which is a classic example you must have studied in your genetics. You cross the plant with a plant that gives you the yellow color flower, which is a recessive phenotype. Therefore, you would expect the genotype to be having allele that are homozygous for the recessive phenotype. So, if you cross, you can see what would be the resulting the F1, what would be the resulting phenotype. You could have two different scenario, assuming the dominant phenotype is because of a heterozygous condition or a homozygous condition for the wild type. You could expect as shown here. So, it will be all F1s either are displaying the dominant phenotype that you would expect if the plant that you have chosen is homozygous for the dominant allele. But you would expect, for example, 50-50 of the progeny showing both the dominant phenotype and recessive phenotype if that plant, the dominant plant, having a genotype which is heterozygous for the dominant allele. So, these are good. Likewise, you can do it for animals and many other species, but it is practically not possible in case of humans, because here this is the marriage is what you call otherwise as a mating. It depends on many other factors, not necessarily for testing anything. So, here normally what we do is we infer from what has happened. You cannot test anything, but you infer from what has happened in the normal course of marriages and mating and so on. So, that is called as pedigree analysis. By talking to an individual of a family and trying to understand who are the other members of the family and who are the members who have a particular phenotype and their history, medical history and so on, who marries to whom, how many children, and this kind of information we put together to construct what is called as a pedigree. So, how does it really help? Let us look into this picture which is represent the very famous family in India, the Nehru Gandhi family. So, you have here two prime ministers, ministers and politicians, everyone in this particular photograph. So, what this picture shows is that you do have important members of the family, but it does not really explain how they are related to each other. If you expand the family, these are the very prominent members of the family. Starts from the first prime minister of the country, Nehru and his wife, and next generation again the most powerful prime minister, Indira Gandhi, her husband Feroz Gandhi, and then the next generation, of course, you have Rajiv Gandhi, who was then a prime minister, Sanjay Gandhi, and his wife and so on. So, this easily you can you see that there are members, but how do you really see in which way they are connected? So, this we do, and by way of connecting them, we will be able to make a pedigree. So, like here, for example, we are shown that Rahul Gandhi, for example, here, the biological fathers for Rahul and Priyanka Gandhi are Sonia Gandhi and Rajiv Gandhi. Likewise, we can go up and so on. We can go up in the hierarchy, in the generation, and you will be able to tell how each one is related to the other. So, this is one way of connecting the family and explaining how they are related to each other. The same thing is done in case of by using certain symbols, we try to represent the different members of the family, their generation, what way they are related and so on, and this is what is shown on the right side. So, you have symbols, we have replaced the symbols with the photographs, again it represents pretty much the same family and we can show. So, this is one way of depicting what is called as a pedigree. Let us see how do you really do that. So, you talk to individuals and construct the pedigree, because you may not be interacting with every member of the previous generation. For example, the great grandparent may not be living now, but still you will be able to talk to their children or their relatives, and still you will be able to understand what kind of phenotype they had, whether they were normal, and what are the other conditions they had. That really helps in constructing a pedigree. So, let us see how do you really do this kind of recording. So, there are symbols, as I said, some of them are already shown. I am going to tell you some of the very important ones. For example, square. The square is a symbol for male, and if you another individual, if you make a circle, that individual represent a female. So, these are the two sex that we represent using symbols, square male, a circle represent a female. At times you do not want to reveal the identity of an individual, or when the someone passed away, even when they were developing in the inside mother, so you do not know the sex. So, that when you are not knowing about the sex, or when you do not want to reveal the sex of an individual, you can use this particular symbol. So, that really does not denote as to what is the sex of that individual. Likewise, if you have this symbol, whether it is square or circle, and if they are unfilled, open, that suggests that they represent the wild type, meaning they are normal. They do not express the phenotype that you are trying to show in that particular pedigree. But on the other hand, if you are filled like this, that represent that these are the individuals who develop or who display the phenotype. The phenotype could be something like what I said, it is closing your, clasping your hands, the left thumb over right, that is the phenotype that you are looking at, or it could be a disease, the affected individual. So, it depends on what you are trying to show in a particular pedigree with regard to how it is being inherited. As I said, there could be multi-generation family. You may have 4 generations in a family, and not all the members of that 4 generation family may be alive today, but you want to even display that in your pedigree. So, you can show that by putting a cross across either the male or female, that particular individual, that would denote that they are no longer alive when you made the recording of the pedigree. So, this is how you do it. There are more symbols. So far what we have discussed on the left side is individuals and their status, but now we are going to show how they are related to each other. Normally when you draw a line between 2 individuals, obviously it would be between a male and a female, that represent the mating, right? They are the biological parents. At times there could be 2 lines like this that would denote that these 2 individuals are related. For example, marriage between cousin. For example, I am marrying, married to my aunt's daughter, for example. That represent what's called as consanguinity. So, that represent consanguinity, suggesting that they are genetically more closer, and this is a marriage within relatives. Now, how do you represent the second generation? So, if you have a line on the top and they are connected on the top, that represent that these 2 individuals are siblings. It could be brothers, it could be sisters, or it could be brother and sister, like what is shown here. You could also have individuals that are twins, they are born at the same time, and they could be of 2 types. Either they are twin brothers, but not identical. They are like pretty much like your otherwise brother, 50% you have shared similarity, and that results because of 2 different embryos growing at the same time. That is why they are different from each other. So, that is denoted by this kind of line, suggesting at the same birth you have 2 individuals, or it could be what is called as twin brother that are identical, resulting from the same embryo. The same embryo was growing, split into 2, and each half has developed into 2 new individuals. They are genetically identical, look alike, and so on, and that is identical brothers or sisters, and that is denoted by a line here, as you see here. Now, you could have conditions wherein there are conditions wherein an embryo is aborted or there is a miscarriage. So, that you denote by a small symbol and cross because it did not survive. So, these are some of the symbols that we use to identify the pedigree. 2 more. This is very important. This you called as index case or proband, because you assuming that you are a genetic counselor and there is a family that comes to you to take some input on on the disorder that may set in the family. So, you get to know of this family because of particular individual who may have a phenotype. So, you talk to that individual, their parents, their brothers, sister, and grandparents, and so on. So, you get to know of the entire family because of one particular person. So, that is denoted by an arrow here. So, that is called as the index case, right. So, or the proband. So, this is how, these are the symbols that we use to show the pedigree or to construct a pedigree. Let us do it now. So, this is, I have shown here now the same Gandhi Nehru family with all the symbols that has been used here. We are showing that you have Jawaharlal Nehru, Kamala Nehru, no longer alive. Next generation is Feroz Gandhi and Indira Gandhi. Indira Gandhi being daughter of Nehru and Kamala Nehru, and again they are not alive now. And you have next generation, Rajiv Gandhi, Sanjay Gandhi, married to Sonia and Maneka Gandhi, and you have a next generation. So easily you can depict, and you could perhaps in your free time you can draw a pedigree for your own family as big as possible. You can combine papers and try to practice. That would really help you even to understand how possibly some of the phenotype that you may see in your family is segregated, which would help in some of the exercise that we are going to do a little later. So what do you really, why should you spend time in making this kind of, diagrams, circles and lines and squares and filling them and crossing them and so on. So this is the way to represent the family. Otherwise, it's, in one go you can understand how each one is related, and you will be able to show a particular phenotype how it is segregated. So in any pedigree, when you draw like this, when you have, for example, the filled one or unfilled one, in a given pedigree, you are going to show only one particular phenotype. Say, for example, you are talking about the handedness, like when you put, when you clasp your hands, the left thumb comes over right. So this is the phenotype. So in a given pedigree, you are going to only, show that particular, trait. You cannot talk about whether the same pedigree, you cannot talk about whether they are able to roll the tongue or not. That becomes complicated. So you may want to use another sheet, but represent the phenotype, but for the same pedigree. You can do that. But traditionally, in a one pedigree chart, you are going to show only one phenotype, because that makes it easier for us to understand how that phenotype is segregating. So how do you really record ancestry? We spoke about the symbols, the other symbol that really explains the relatedness, in what way they are related. But we also have to identify an individual. So you cannot say again using a relation. So one simple way to show it is, on the left side you use Roman numerals, identify the generation, and use the your Arabic numerals to identify the individuals, right? Like for example, second generation, individual 3, 4 and so on, third generation and so on. So it is easy for you to refer to which individual you are talking about, right? So in each of this you can see that this individual you call it as married in, because she is not related genetically with the family, but she is married to this individual who is related to the previous generation. Likewise here, but you denote that, right? So that's how you do it. So you really build the pedigree this way. So these are thumb rules, because you follow it, because anyone else again can look into your pedigree, can understand what really you are trying to explain. So these are standard symbols. Let us try to draw a pedigree for a phenotype that we have already discussed, that is polydactyly. We said it is a dominant disorder, meaning you may have a sixth finger, it is because of a defect in one of the genes, and it is a dominant phenotype. If I have the defect, I am going to have a sixth finger, right? So, but it is abnormal because it is not seen in the wild type. All of us, majority of us have 5 fingers in our hands, but certain individuals, extremely rare condition, you do have sixth finger, right? So how do you, assuming that I am an individual who has got sixth finger, I am going to draw my pedigree. So what I will do, I will interview myself. Let us assume that I am here. So I am just, this is me, and I am starting the entire family with myself, being the index or proband. So I have the the sixth finger. So I am the individual who is affected, right? I express the phenotype. So how do I really build my family? I first ask how many brothers I have. So I am fourth in my family. I have 2 brothers, 1 sister. So I construct, none of them have. They are all normal with 5 fingers, right? And of course, I am talking about my parents. So it is a dominant disorder. Obviously, I should have gotten the bad gene from one of my parents, right? So assuming my mother is also having sixth finger, right? So I would talk to my mother whether in her family anyone else had this, and I understand and her brother also had 6 finger. Then I asked whether her parents had. It looks like, my grandfather also had 6 finger, right? And his sister, that is my 2 generation back, you can talk about that way, she also had. So you can go on building a pedigree like this by talking to people, right? So this would explain as to how, from where possibly the gene that gave you this particular phenotype, that is having 6th finger, came in, right? So that's the one way to construct the family, right? The question is, how do you, by doing this, what really, how does it help you? So one immediate benefit, or not immediate benefit, one of the benefits of this pedigree analysis is that you would understand as to on which of the, the chromosome you have the defect that results in, for example, 6 finger. The pedigree analysis really helps you to start with this gene hunt as to where it is. Let's see, so how does it really help? Let's assume that this is the family, right? One of the families that we are talking about, and you call it as this inheritance here, you call it as autosomal dominant. You call it as autosome because you assume the defect in a gene that results in this phenotype is present in one of the chromosomes that is autosome, meaning every other chromosome other than X and Y, right, the 22 pairs. And you call it dominant because you would, in the model, you would anticipate even if one of the 2 copies of the gene that you have is defective, you would have the phenotype. That is why you call it as a dominant phenotype, right. Let us see, this is a pedigree that is shown. Let us see how does it really help us to tell whether it is, the defect is on an autosome and the phenotype that you see is dominant, because you really do not know where is the gene, you do not know where is the mutation, you do not know what kind of mutation, nothing. You only spoke to that individual, and let us assume that individual happened to be, somebody here, let us say your friend, and that is how you came to know about the entire family, and you sort of plotted this, and you want to ask what is the mode of inheritance. In an autosomal dominant inheritance, what you would see is that an affected person usually has at least one affected parent. Go with the model, you should have inherited at least one defective allele from one of the parents. Even one is good enough to show the phenotype. Therefore, you can see here that her mother is affected. Likewise, you take any individual, for example, his or her father is affected. Likewise, her mother is affected, and so on. And likewise, if you look into this, each one of that here also, you find that one of the parents of each one of the affected individual here is also affected. That is what you are saying. Affected person usually has at least one affected parents. Affects either sex, because autosomes are present in both male and female. And likewise, is transmitted by either sex. You can see that male, female both are affected, transmitted by either by a female or by a male. Both are possible because it is autosome. And what is the probability? If a child with one affected and one unaffected parents, has, for the next child, it is 50% chance being affected, simply because of the 2 parents, one is having the mutant allele in a heterozygous condition. So there is a 50% probability each gamete of that individual carrying the affected allele. That is why it is 50% probability of having the affected phenotype. So this is for autosomal dominant inheritance. Let us look into how the genotype would look like. This I just try to plot it over there. You can see here, you say dominant because this individual would express the phenotype even if one of the allele is mutated, and likewise you can see everyone here, you are having an allele which is the dominant, which is shown here with the capital A, and that allele is present in every affected individual, and you can show that dominant allele, that capital A, has come from affected parent, right. So that's how it is. You can really trace it. So this is the model that you can use it to identify or even predict that a given phenotype is dominant phenotype. Let us look into an example of how autosomal recessive inheritance might look like. Here is a pedigree that represent a phenotype resulting from autosomal recessive condition, and as compared to the dominant condition, you may have a generation in which the individual may not show the phenotype, right, the reason being given here. So, usually if you look into a pedigree that is autosomal recessive character being inherited, you will find that affected people are usually born to unaffected parents, and more often you would find consanguinity in parents. Like for example, here you are showing two lines which because they are related, because they share the same ancestry, you can see here. So, that is what is shown here, and parents of the affected people are usually asymptomatic. Likewise, you see here, this is a affected individual, their parents do not show, but in the previous generation you may have an individual who is showing the phenotype. There is an increased incidence of parental consanguinity as depicted here. Again, it would affect either sex because it is autosomal. The gene that is causing the disorder or defect in the gene causing the disorder is located on one of the 22 autosomes. After the birth, we are talking about, say suppose this individual had the phenotype, and if they are planning for one more child, what is the probability that that child, the fifth one for example, would have the same phenotype. So, after the birth of an affected child, each subsequent child has 25% chance, because for you to have the phenotype, you have to have both alleles carrying the mutant forms of the gene, then only it would show. And if you could recollect the Mendelian Punnett square, like what is the, like if you plot it, then you would find it is 1:3. So, that is the kind of that is why it is 25%, right. We will come back to that a little later. So, that is the example of how autosomal recessive condition would be transmitted in a disease. Just to plot the genotype, therefore, you can more easily understand. And we talk about consanguinity, here you see that this is the consanguinity between, this is a mating or marriage between relatives, and both the individuals here are heterozygous for the mutant allele. Let us assume the small a resulting in the phenotype, and this small a allele is contributed by both the parent, therefore, you have homozygosity, the individual is showing the phenotype. And if you go back to their parents too were heterozygous for the mutant allele, and this mutant allele actually came from their parents, both of them are heterozygous, and you can say in this individual, unfortunately the mutant allele contribute to the phenotype. So, from here on the mutant allele co-segregates in the family, and given a chance they may have the phenotype. So, that is an example of autosomal recessive condition. That is the example for autosomal recessive condition. So, let us go beyond the autosomes. So, with this kind of pedigree analysis, you will be able to tell whether a gene or a defect in a given gene results in a dominant or recessive condition, and the gene is located on one of the 22 chromosomes, which you call as autosomes, right. But how would a phenotype look if the gene, defective gene, is located on this particular pair, which you call as sex chromosomes. So, these are their distribution or their segregation, their expression is very, very different. There are disorders as well associated with the sex chromosomes. For example, hemophilia, the blood clotting disease, and red green color blindness, these are the genes for this particular phenotype is located on the X chromosome. So, let us look into how a phenotype linked to X chromosome segregates in a family, right. There is a complication when you talk about X chromosomes and their expression of the phenotype. As we discussed in the central dogma and also about the chromosomes, we have male and female. The sex chromosome composition is unique to each sex. In female, for example, here you have 2 X chromosomes, there is no Y, whereas male have got 1 X chromosome and 1 Y chromosome. So, depending on whether a mutant allele is present on XY individual, that is male, or XX individual, that is female, you may or may not show a phenotype. That is what denoted here. For example, let us assume that you are looking at a phenotype resulting from a recessive condition, meaning both the alleles should carry the mutant allele, and then only you would have a phenotype. Both copies of the chromosome should carry the mutant allele, then only you will have the phenotype. In case of female, you may you may have a condition wherein you have one X chromosome which has got a normal in a wild type allele, other one is having the mutant allele, and the female may not express the phenotype because she carries a normal allele there. But in case of male, because you have only one X chromosome, if that X chromosome carries the mutant allele, invariably the male would express the phenotype even if only one allele is present, right. But there is one more complication. The female who is carrying only one mutant allele at times may express a phenotype because of the phenomenon called as X inactivation, because there are two X chromosomes in female, the number of the copies of the gene that are there in a female also is twice as the number of gene copies that are there in the male. To compensate the dosage, one of the two X chromosome gets silenced, and this silencing is random. So, in certain cells, for example, X chromosome 2 gets inactive, in certain cells X chromosome 1 gets inactive. So, it may be a chance that in majority it could be X chromosome 2 which carries the wild type allele may be inactive. As a result, you express a mutant allele and you may end up having the phenotype even though you are heterozygous and even though if the phenotype is recessive. So, this could happen. So, that is the complication that comes along with X chromosome. Even then, since the inactivation is random, you assume that in 50% of the cells it could be inactive and so on, still you will be able to trace whether the phenotype expression of a X-linked gene is recessive or dominant, still you will be able to do it. So, let us look into recessive condition, right. So, that is what is shown here. This pedigree represent an autosomal X-linked recessive phenotype, and you can see here that you have introduced a new symbol, that is you have a dot inside an individual, like either it is a circle or at times it could be a square, that denotes what is called as a carrier. So, that means that these are asymptomatic individuals, but do carry the defective allele. So, that is what it denotes. Why do you say that? Because of this model, that is it is X-linked recessive condition. Let us see, like. So, if you have a recessive condition for X-linked disease, more often it would affect the male, because in female the other X would have a normal copy of the gene, therefore, you may not express. But invariably in case of male, if that individual somehow received the X chromosome which has got the defective allele, he would end up showing the phenotype, because there is only Y chromosome that does not really complement the X. So, therefore, it you will find the majority of the times in such families it is the male who is affected. The second is that affected males are usually born to unaffected parents, because the X's come from mother. The father will not transmit X chromosome to son, then that individual becomes daughter, because if son has to be born, then father should have given the Y chromosome. So, it does not come. So, it comes through mother, and the mother has got the other X chromosome which could be normal, therefore, she may not show a symptom, but she is a carrier, and you can see it here. For example, here the son is affected, his mother is a carrier, but asymptomatic, not showing. Likewise, her mother is asymptomatic, but carrying the defective allele. Likewise, her mother is a carrier, not showing symptoms. The mother is normally an asymptomatic carrier and may have affected male relatives, right. So, likewise, you can see here, you can go back in the family, you would find some of the males are affected who are related to the mother, because that if that X chromosome results in a male, then certainly he would show phenotype, because he does not have Y chromosome, he does not have the other copy, wild type copy. Females may be affected if the father is affected and the mother is a carrier, right. It can be transmitted by a father to a daughter, right, that is like what you can see here. But if the mother also contributes X chromosome, if she is a carrier, then the daughter also can be affected, right. Occasionally that happens, and as I told you, there is a phenomenon called inactivation, X inactivation. At times it could be skewed, it could be non-random, meaning in majority of the cells, the allele that is having the wild type allele, that chromosome may be inactive. Therefore, you would, even though it is a recessive condition, if you are heterozygous, still you would show the phenotype because of the abnormal X inactivation, right. So, what is important here is that in this kind of inheritance, you will not find a male to male transmission. In any of the generation, you will not find a male transmitting the phenotype to a male in the next generation. That is not possible because it is there in the X chromosome, right. So, that is, but you look into the phenotype, it does not segregate, then you can call that the whatever phenotype that you are looking at could be X linked recessive. So, you use these thumb rules to understand whether the phenotype could be X linked or autosomal, whether it is dominant, recessive. So, you sort of come up with an hypothesis, and if you can apply that in multiple families having the same genotype, it becomes more and more clear that this is indeed the mode of inheritance. That is how you do it. Let us look into the other condition, that is X linked dominant condition. Even if one allele is defective, so you would show the phenotype in case of female, right. So, here it affects either sex, but more females than males. Why? Obviously, if it is a female, then she is going to give this X chromosome to 2 individuals in next generation, right. So, therefore, from female it will go to more number of individuals, therefore, you would have more of them, and more often it would be females, right. So, that is what it is. Usually at least one parent is affected, because the dominant condition you would expect either father or mother, both of them could show the phenotype. But females are often more mildly or more variably affected than males because of a difference, that is the number of cells in which the given X is inactive. So, because again there is X inactivation condition, so you will have a variability in terms of how severe the phenotype if it is a disease. The child of an affected female, regardless of its sex, has 50% of chance being affected, because she has got 2 X chromosome in her meiosis, she is going to gift, if she is a carrier, heterozygous, 50% of our germ cells would have the affected chromosome, therefore it is 50% chance. So, an affected male, all his daughter but none of his son are affected, because the moment he contributes X chromosomes in his sperm, that sperm, whichever egg it fertilizes, that would result in a daughter, right, not in son. So, therefore, the daughter will be affected if it is X linked. So, this is how you really look into the family and come up with a hypothesis or a suggestion that the phenotype that you are seeing is a dominant, recessive, and what it is. So, let us look into the other chromosome, sex chromosome, that is Y chromosome. It is very, very interesting, because the Y chromosome is the smallest chromosome in your cell, and it does not carry all the essential genes for a normal, which is the function that are required for both male and female, because the female do not have Y chromosomes, still they are normal. So, it has genes that contribute to the maleness, like developing into a male, the behavior, the fertility, the sperm functions, and so on. Therefore, really you do not see any disorder that linked to Y chromosome that runs in the family, because if there are defect in Y chromosome, the male embryo will not form, or if it is defect in Y chromosome, the fertility will be affected. So, it does not run in the family. So, therefore, you do not see disorders that linked to Y chromosome. So, it is an hypothetical family. If there is a phenotype linked to Y chromosome, this is how it will look like. So, it would be always from a male to male to male, right. It does not go the other way, right. It would affect only the males. Affected males always have an unaffected father, because they have gotten the Y from their father. All sons of an affected man are affected. So, these are the thumb rules you would use, but hardly there are any condition that runs in family which are linked to the Y chromosome. So, that is pretty much the condition. This is what is shown here, like you have Y which is having a defective allele, how it would be transmitted in the family, right. So, this is for all the genes that are present on your genome, the 23 pairs of the chromosome, autosomes and X chromosomes. But we also have DNA in one of the organelle, that is mitochondria, and its mitochondria is very, very important for your function of the cells, because they provide energy, and mitochondria has its own DNA, and the DNA, if it is defective, you could have condition. There are conditions that affects there are muscular dystrophies, there are neurological disorders resulting from defect in mitochondrial DNA. So, therefore, and then mitochondrial DNA is also transmitted. You receive mitochondrial DNA from your parents. So, if the DNA is defective, how it would be transmitted, right? That is one particular question you are trying to address here, because the phenotype is very unique. The segregation rather is very unique. So, that is what we are seeing here. So, there is a family that is drawn, pedigree, and we are looking at a mitochondrial disorder, and you see that a large number of them are female affected, right. Let us look into that. There is a unique thing about mitochondrial DNA, because the mitochondrial DNA that you receive it invariably from, whether you are a male, whether you are a son, you are a daughter, you receive the mitochondrial DNA from your mother. The reason being, you have a cell which is the egg, which is yet to be fertilized, and it carries the mitochondria, which carries the mitochondrial DNA, which is circular, and it has got certain genes which are transcribed, makes protein that are required for mitochondrial function. Now, what is interesting is that when the egg and the sperm fuse to form what is called as a fertilized egg or zygote, the interesting aspect is that the mitochondria of the sperm are not transmitted to the embryo, so the fertilized egg. So, it is only the mitochondria of the mother that is retained in the egg. Only the nucleus that carries the DNA is allowed to fuse with the egg. Therefore, if you are a male, you do not contribute your mitochondrial DNA to the next generation. It is invariably it is the mother who contributes, right. So, any defect that if you have developed in your mitochondrial DNA, that will not go to the next generation. You may receive a defective DNA from your mother, but you will not contribute that or segregate that to the next generation. Therefore, the inheritance pattern of this particular phenotype resulting from mitochondrial DNA defect is going to be very different. So, that is what, how do you really analyze that? It affects both sexes, because mother contributes to both male and female, and the egg is anyway is given by mother, and whether it would develop into a male or female depends on the sperm, but sperm does not contribute to the mitochondrial DNA. It is usually inherited from unaffected affected mother. Mother is carrier, or she is showing the symptoms for particular disorder, can be caused by de novo mutation, meaning the mutation could have come in the egg itself. For example, the egg somehow had a mitochondria which had some defect, and therefore, that embryo developed. This is called the de novo, meaning a new mutation coming in a germ cells, right. It is not transmitted by father, as I told you, father does not contribute to the mitochondrial that is there in the embryo. These are highly variable clinical manifestation. Reason being, you could have difference in terms of the number of mitochondria that is having the defective DNA. If majority of the mitochondria has got defective DNA, you are going to show a phenotype which could be severe. Only if it is 10% of the DNA or 10% of the mitochondria carries the bad DNA, your symptoms could be very mild. So, you would have variations in the manifestation. That is what called as heteroplasmic. That is the term that is shown on the top, right. And it is called the matrilineal inheritance, meaning it is always transmitted by mother, and frequent heteroplasmy, meaning there is a variation, right. It is not that every mitochondria in a cell is defective, and it varies. When the cell divides, how the mitochondria are distributed to the two daughter cells, that determines whether the cell would function normally or not. If so happened one of the two daughter cells inherited most of the bad mitochondria having bad DNA, that cell may not function as good as the other cell which is lucky enough to get most of the good mitochondria. So, how severe the phenotype depends on how what is the burden of that cell in terms of how much of the bad mitochondria, how much of the bad DNA that it inherited, right. So, that is why you show or you see a variation, right. So, these two, that the Y chromosome which is transmitted by male to son, likewise the mitochondrial DNA transmitted by mother to next generation, which only runs through one of the two sexes, is really is very interesting. That helps people to understand how we evolved, right. So this is this is something which I I really recommend all of you to go to this particular blog written by Denise Venema. She has written a beautiful blog on how Y chromosomes and X chromosome really the DNA sequence there we can use to analyze and understand how we evolved, right? So we can really talk about what could be the mitochondrial sequence or the genome sequence, from where we evolved. We talk about we all evolved from Africa. So this is it a myth or is it a reality? Is it possible? Can we scientifically prove this? And people have used these two DNA material to strengthen the hypothesis that all of us have evolved from a common ancestor somewhere in Africa, right? So why it is so easy to test this hypothesis using Y and Y chromosome and mitochondria? Because of this fact. For example, you have different mitochondria, meaning they have a difference in the DNA sequence, right? And 3 individuals, and they keep marrying, and you have a large population. By looking at the sequence difference, you will be able to really trace who could have been the original mother who seeded the mitochondria. So it's easy for us to see, because always the mother contributes the mitochondria to the next generation. Likewise, we can use the Y chromosome or sequence therein, and which could be, for example, individual 1, individual 2, individual 3. There are variations in the Y chromosome gene sequence, and this sequence we can really trace it, and we will be able to understand the ancestry, who are the original fathers, and how the population has mixed. And this is possible only if you have traced the mitochondria and Y chromosome. But if you have, because the segregation is either from father or from mother. However, if you are trying to trace, for example, an autosomal gene, it's very difficult, because you cannot trace now, all sort of mixture takes place. It will be very difficult to understand whether it has come from one of the 2 parents or both of them, and so on. So it is extremely difficult. So most of the studies where people to try to understand the population dynamics or population migration or population evolution, they have looked at the sequence difference in Y chromosome and mitochondria and try to link and come up with hypothesis. And one of the studies that came from India by a group at Hyderabad really used this mitochondria and Y chromosome sequences to support and come up with an hypothesis which suggests that our forefathers evolved around 200,000 years ago somewhere here, northern Africa, and they probably migrated close to the seashore. That this is people thought that they straight went like this, but what we understand, probably they went through the sea route. They did not go through the land route, but they probably went through the sea route to the other parts of the world. This is how now the current hypothesis is. It all because of our understanding of how the mitochondrial DNA sequence and how they are transmitted, and how the Y chromosome is transmitted, what are the sequence variations. We can use this to even go beyond 3 generation, 4 generation, maybe hundreds of generations back, and we will be able to tell how related we are with in relation to the other races in the world, and so on. So that's the power of genetics, and that's what we pretty much looked at in this particular chapter, right? We'll see in the next week.